Good morning, class. This is Dr. Pratibha K.S., Head Department of Biotechnology at KL Society's S. Nilingappa College, Rajayanagar, Bengaluru. Today, I'm before you to present Bengaluru City University syllabus for BSc Biotechnology UG course. Here you are studying choice-based credit system, that is CBCS. I'm discussing before you a first year biotechnology paper. Okay, undergraduate program framed according to the National Education Policy, that is NEP 2020, and this is accepted from the academic year 2021 and 2022 students. And this is our Bengaluru City University logo. Okay, here in Kannada it is written Aguni Aniketana. Okay, be boundless. Now let's go through the syllabus of Bengaluru City University Biotechnology course. Okay, model curriculum. We are going through name of the degree program BSc Basics or Honors Discipline Core Biotechnology. Total credits for the program BSc Honors is 176, and which we started from the year 2021 and 22 we have implemented. Okay, so we can go through all these things in the website. Further, let us go through the course. Okay, here progressive certificate, diploma, bachelor's degree or bachelor's degree with honors provided at the end of each year of exit of the 4 years undergraduate program. You are studying now. First year we'll call that as certificate course, then diploma course, then basic bachelor's degree, then fourth year will be bachelor's degree with honors. Okay, so first year when you finish your first sem and second semester, that will be called certificate course. Okay, 2 semester, and if you completed 4 semester from the BCU, BSc course, okay, diploma course, okay, then 6 semester when you finish, you will be called basics bachelor's degree. Then if you finish 8 semester, then you will be called bachelor's degree with honors. And here you could see different credits. You just keep in your mind first year certificate course, then second diploma course, then third basic bachelor's degree, then fourth one is bachelor's degree with honors. Okay, now let's go through the syllabus. So you're studying right now semester 1, and you're studying theory paper discipline T1 BTC 101A1 cell biology and genetics. Okay, in a week 4 hours theory class will be there, and you are studying practical, that is again cell biology and genetics. Open elective we have biotechnology, that is biotechnology for human welfare. Okay, so total credits 25. Now let's go through our basic content of the particular course. So here this is BSc Biotechnology honors, basic or honors first semester. So content course is point 1 theory DSc T1 BTC 0101 cell biology and genetics, which is 56 hours. Unit 1 is cell and cellular organelles, total hours 14 hours. Under this cell and cellular organelles, you are studying historical perspectives, discovery of cell, the cell theory, ultrastructure of a eukaryotic cell, both plant and animal cells, structural organization and functions of cell wall and plasma membrane. Further, structure and functions of cell organelles like cytosol, endoplasmic reticulum, Golgi complex, mitochondria, chloroplast, ribosomes, lysosomes, peroxisomes, nucleus, nucleolus, vacuole, and cytoskeletal structures like microtubules, microfilaments, and intermediate filaments. Okay, all this you are studying under Unit 1, Cell and Cellular Organelles. Then Unit 2, Chromosomes and Cell Division. General Introduction, Discovery, Morphology and Structural Organization. In that, centromere, secondary constriction, telomere, chromonomer, euchromatin, and heterochromatin. Further, chemical composition and karyotype. Okay. Single standard and multi-standard hypothesis, folded fiber and nucleosome models. Apart from all this, you are studying special type of microorganisms like salivary gland chromosome and lamp brush chromosomes. Okay, cell cycle, phases of cell division, mitosis and meiosis, cell cycle checkpoints, cell cycle checkpoints, enzymes involved in regulation, cell signaling, cell communication, significance of cell cycle, achromatic apparatus, synoptimal complex, senescence, and programmed cell death. So unit 2, again, it's for 14 hours. Next, unit 3, inheritance and gene interaction. Here you are studying history of genetics, Mendelian theory, loss of inheritance like dominance, segregation, incomplete dominance, co-dominance with an example. Further, law of independent assortment, test cross, back cross, and non-Mendelian inheritance. Okay, so all this comes under history of genetics. Then maternal inheritance. Here you are focusing on plastid inheritance in Mirabilis jalapa, kappa particles in paramecium, and pitted characters in yeast. Further, sex-linked inheritance, chromosome theory of inheritance. Okay, next year, gene interaction, supplementary factors, comb pattern in fowls, complementary genes, flower color in sweet peas, multiple factors, skin color in human beings, epistasis, plumage color in poultry, multiple allelism, example, blood groups in human beings. Okay, and this unit 3 also carries 14 hours. Then further last unit, unit 4, linkage and mutation. Here you are studying general introduction related to linkage. Okay, then coupling and repulsion hypothesis, linkage in maize and Drosophila, mechanism of crossing over and its importance, chromosome mapping, linkage map in maize. Next, mutations, types of mutations like spontaneous and induced mutagens, physical and chemical mutagens, mutation at the molecular level, mutations in plants, animals, and microbes, and also its merits and demerits. Okay. Further, structural and numerical chromosomal aberrations, sex determination in plants and animals, concept of allosomes and autosomes. Okay, and you are going to study about the crosses like XX, XY, XS, XO, ZWO, ZZ, ZO, ZZ types. Next, allosomal, that is Klinefelter syndrome and Turner's syndrome. Okay, then autosomal syndromes like Down syndrome and Cray-Duchat syndrome. So all these conditions, you are going to study the clinical characteristics, okay, and their symptoms and treatment you are going to study under this particular allosomal and autosomal disorder. So this is all about your first semester theory part, which comes under cell biology and genetics first semester. All right. Next, let us go through the internal assessment segregation. Okay, so formative assessment, pedagogy, lectures. We can take all the your classes in the form of lectures or PPT presentations or videos, assignments. Okay, we can provide, and weekly formative assessment test can also be given. Okay, assignments or field visit or project which carries 15 IA marks. This column is for weightage in marks, and this is assessment occasion. Okay, and test which carries 20 marks, participation in class in different activities at different platform which carries 5 marks. So total you have internal assessment marks 40 marks. Okay, this 40 marks is like internal quality of the student. So you need to focus on all these things and you need to value your marks. Then further, let us go through cell biology and genetics laboratory content. Okay, here this course content 01 practicals DSc P1 BTC 101 cell biology and genetics. Okay, this is the course title, cell biology and genetics. Course credits 2. Course code DSc P1BT101. Okay, and per week we need to take 4 hours class. Total contact hours 28, duration of your exam 3 hours, formative assessment marks 25. So here 40 marks from your theory and from your internal 25 marks. So 65 marks, this is 40, 65 marks is your internal assessment. Now let us focus on the content, a list of experiment what you need to perform in your practical. Okay, operation and working principle of simple and compound microscope. First and foremost important thing, basics, you are going to understand the working principle and how this compound and simple microscope work, their operation, SOP, standard operating procedure you are learning, then working principle you are going to understand. Okay, then use of micrometry, measurement of onion epidermal cells and yeast. Then study of mitosis in onion root tips. Then study of meiosis in grasshopper testes or onion or rio flower buds. So these are different samples we can use. Any one sample can be used during the practical. Then mounting of polyethylene chromosomes, buccal smear, Bohr bodies, karyotype analysis like human, normal and abnormal. Okay, and onion. Normal male and normal female. Abnormal, you can observe here autosomal syndromes and allosomal syndromes. Eighth experiment, isolation and staining of mitochondria are chloroplast. Ninth one, enumeration of RBC using hemocytometer. Okay. Further, simple genetic problems based on gene interaction. Okay. Last experiment, preparation and submission of 5 permanent slides of mitosis and meiosis by each student. And this carries 2.5 marks in your practical scheme. Okay. So next, pedagogy. In this particular practical 25 IA marks. Okay, here assessment or assignment or monograph that carries 10. I have taken this assignment in consideration. So for your assignment submission in practical, this carries 10 marks. Then test carries 10 marks. Preparation in a class, in your practical class, your observation book and how you perform, observe everything, that carries 5 marks. So total practical IA marks are 25 marks. So from theory, how much marks? Yes, right. 60. Sorry, no, sorry. Total 65. From theory, you have IA marks is 40. From theory, 40 IA marks, and from practical, 25 marks. So total 65 marks is your IA marks in your first semester. Okay, and here there are some reference books you can refer. Okay, you can take screenshot, and if you want, you can refer all these things. And anyway, we are giving notes also, you can refer those notes, or else you want, if you want to refer any kind of books, you can refer Benjamin Levin of different volumes. You are going to get gene second 3 5 4. Okay, cell biology ELBS publications you can refer. Okay, and other book like molecular biology of cell you can refer, genes and genomes. Okay, then molecular biology. Okay, then some other books there are, just go through this all these books. Okay, don't purchase all these books. Most of the books you'll find in the library, you can refer in the library, or even you can refer online content. And even almost all whatever the notes we are preparing, that is prepared by using different authors book, you can refer that also. And if you want any books, you can go to this one. Even one more, you can refer cell biology and genetics, Varma and Agarwal. Very basic book. You can refer that also. So this is all about cell biology and genetics, practical list of experiments, and IA marks is 25. Okay, then this is your theory portion. Okay, if you want, you can take screenshot and kindly make a note in your notebook. The first page of the notebook, kindly write all these. Write, don't take printout. Okay, don't do that any kind of copy paste wala thing. You please write each and every syllabus in your first page of your notebook, so that once a teacher complete these particular units, you can just tick that one. Okay, so I hope you understood today's first semester uh, theory syllabus and practical syllabus. If you have any doubts, feel free to approach. Okay, so this is Dr. Pratibha K signing out for the day. Stay tuned with me. I'll be back with some more videos. Thank you everyone for watching this video.